BioClock Studio Winter 2020 presents their Circadian Transcriptomics Dry Lab tutorial, Part 1 of 4, Introduction to Circadian Omics and Applications. Most organisms have an endogenous 24-hour circadian clock that guides physiology and metabolism according to the time of day. Circadian misalignment, or a state in which the body's endogenous clock is out of sync with environmental or behavioral cycles, is a risk factor for a plethora of illnesses, such as cancer, mood disorders, diabetes, and cardiovascular diseases. To better understand circadian alignment and misalignment, circadian biologists have employed omics approaches to identify and analyze patterns of the expression of genes, proteins, and metabolites that are controlled by the circadian clock. This type of approach allows scientists to bridge the gap between genetics and clinical phenotypes. What exactly is an omics approach? Omics refers to the sampling of all molecules of a certain class in a sample at once. There are many kinds of omics studies. Transcriptomics is the study of the transcriptome, the complete set of RNA transcripts that are present in a particular sample. Transcriptomics has enabled scientists to identify differentially expressed genes and then assess how these genes correlate to phenotype or behavior. Likewise, metabolomics is the study of all the metabolites, and proteomics is the study of all the proteins that are present in a cell or tissue sample. For example, Dr. Amandine Sheks, a scientist at the Salk Institute, and her team have employed transcriptome and metabolome analyses to test the hypothesis that time-restricted feeding, or TRF, can prevent obesity and metabolic syndrome in mice that lack a circadian clock. The rhythmic liver transcriptome, that is, the subset of genes that are expressed rhythmically across the 24-hour day in the liver, includes many genes that encode proteins that are important for a variety of metabolic functions, such as absorption and breakdown of carbohydrates, fats, and proteins. The expression of many genes encoding key metabolic regulators have robust circadian rhythms in mice fed a normal diet ad libitum, which is a term for always available. Under these conditions, the mice consume about 85% of their calories at night, which is their active period. However, mice fed a high-fat diet ad libitum eat throughout the day and night, become obese, and have damped rhythms of these metabolic genes. The two situations lead to healthy mice fed normal chow and mice with adverse health profiles on the high-fat diet. When male mice are given a calorically equivalent amount of high-fat food over an 8 to 12-hour period during the night instead of freely throughout the 24-hour day, known as time-restricted feeding, or TRF, rhythms of feeding and metabolic gene expression are restored. Additionally, TRF is effective in preventing and even reversing many metabolic disorders that can be caused by changes in metabolic gene expression in diet-induced obese mice. This correlation between improved circadian rhythmicity under TRF, as well as its observed health benefits, led Sheks and her colleagues to ask, does the circadian clock drive feeding rhythms, which in turn drive rhythms in the expression of metabolic genes? Or does the molecular clock itself activate genes to drive daily changes in metabolism? Can TRF compensate for the absence of a molecular clock by imposing a feeding rhythm? To test this hypothesis, they used dietary and genetic interventions in mice and used genomic and metabolomic analyses to examine the genes, proteins, and metabolites responsible for the phenotypes they observed. They subjected clock mutant mice, both whole-body Cry1, Cry2 knockout mice, and liver-specific BMAL1 and reverb alpha-beta knockout mice, to a high-fat diet under one of two feeding paradigms, time-restricted feeding or ad libitum feeding. Both groups consumed the same amount of food and number of calories, but the TRF group was given access to the food only during a 9 to 10 hour window during the dark phase, when nocturnal mice are normally awake and active. After 12 weeks, liver samples were collected every 4 hours over a 24-hour period for wild-type and liver-specific BMAL1 knockout mice, and every 3 hours over a 24-hour period for liver-specific reverb alpha-beta and Cry double knockout mice. The samples were used for transcriptomic and metabolomic analyses. Why some every 4 hours while others every 3 hours? It is likely that in the case of this study it was due to practical limitations, such as the number of mice available. But it is interesting to consider how the sampling resolution, or the interval at which samples are taken in a 24-hour period, affects the ability to detect cycling genes. A study by Dr. John Hoganesh and colleagues showed that an increase in sampling frequency, that is, collecting samples at more frequent intervals over 24 hours, results in an increased number of cycling genes that can be detected. Therefore, the wet lab experimental considerations affect the results generated by dry lab analysis. To gain insight into the molecular pathways that are regulated by TRF, the researchers performed metabolomic and transcriptomic analyses on the liver samples. The effect of TRF was analyzed within or across genotypes, and cycling genes and metabolites in all groups were identified using a statistical tool called Metacycle. First, the researchers compared the effect of TRF, or ad libitum feeding, on the clock-deficient mice. After 12 weeks, the clock-deficient mice under the ad libitum schedule had negative metabolic consequences, including excessive weight gain and features of metabolic disease such as fatty liver, high blood lipid levels, and glucose intolerance. However, the clock-deficient mice on the TRF schedule were largely protected from these adverse outcomes. That is, they had healthier measures by most criteria. So when a feeding and fasting schedule is imposed on the clock-deficient mice through TRF, they maintain good metabolic health. These findings suggest that the circadian clock maintains metabolic homeostasis by providing a consistent daily cycle of feeding and fasting, and this cycling can be imposed externally. The development of metabolic diseases in the ad libitum-fed clock-deficient mice is largely a secondary consequence of their behavioral disruptions, not solely caused by disrupted clocks. These findings show improved feeding and fasting rhythms and associated metabolic oscillations can override faulty clocks and improve metabolic health.